Carol Devamani recently completed a research project in rural India on hand washing as part of her studies at the London School of Hygiene and Tropical Medicine. Carol, could you please describe the study?
We included 14 mothers in 4 different villages in Andhra Pradesh, and we looked at the amount of bacteria on the hands before and after hand washing with soap at regular intervals till the 1.5 hour mark.
Could you describe the methodology used?
We used the direct finger impression technique on special agar plates, and this is where 4 fingers and the fingertips specifically are placed onto the agar plate, followed by the thumb impression. And so 1/2 of the agar plate is for one hand, and the other half is for the other hand.
What bacteria did you identify?
We looked specifically at Enterococcus and E. coli, which are fecal indicator bacteria, and these 2 bacteria, when present on the hands, point out poor hand hygiene. And when it grows on this special agar plate, it takes up a specific color. So Enterococcus took up the turquoise color, and it's a pinpoint colony, whereas the E. coli takes up this burgundy color, and it's slightly larger in size.
Could you show us the agar plates from the different time intervals and the graphs with your findings?
So this first plate is taken just before hand washing with soap, and here there's a lot of bacterial growth, as indicated by the number of colors that one can see. But the next plate is immediately after hand washing with soap, and here the growth has come down quite drastically, and the colors as well cannot be seen as before. And at the half-hour point following hand washing with soap, once again the growth has come back up. You have a larger number of bacterial colonies, and you can see that by the different colors. When we put this into a graph, a trend can be seen, and that is before and after hand washing, this V-shaped sort of trend. So before, there's a high level prior to hand washing. After hand washing, it drops, so there's this sharp drop, followed by an increase once again.
What do these results mean for hygiene promotion and sanitation?
So right off, it's clear that recontamination rates are high and very quick, and in this setting it's understood and could be explained largely due to environmental exposure, namely because of contact with animals and practices of open defecation. So hand washing alone may not be enough in this kind of setting, and so better sanitation could decrease the amount of contamination in the environment. So one needs to focus both on hygiene and sanitation simultaneously in order to improve health.